And welcome to the second Tiger in STEM chemistry webinar series for summer 2021. My name is Dr. Sean Collins. I'm at the School of Chemistry and the School of Chemical and Process Engineering at the University of Leeds. I'd like to just jump right in and introduce our chair for this session. This is Professor Karen Folds. Karen Folds is a professor in the Department of Pure and Applied Chemistry at the University of Strathclyde and an expert in the development of surface-enhanced Raman scattering, or SERS, and Raman techniques for novel analytical detection strategies, and in particular, multiplexed bioanalytical applications. She is a fellow of the Royal Society of Chemistry, the Society for Applied Spectroscopy, and the Royal Society of Edinburgh. And she was the first woman elected chair of the Infrared and Raman Discussion Group, IRDG, in 2014, which is the oldest spectroscopic discussion society in the UK. Thank you very much for chairing this webinar today. I'll hand it over to you now. Thanks.
Thank you, Sean, for that kind introduction, and thank you very much to the Tigers in STEM for inviting me to chair this session. I'm really impressed with all the great things that they're doing for equality and inclusivity in STEM, so I'm really impressed by this great lineup of speakers that we have this afternoon, so I'm really looking forward to the next hour. I've got some housekeeping that I need to pass on to you, so please bear with me as I try to remember all the things that I have to say. So we should start by noting that there's a code of conduct for this event. Someone will post a link to this in the chat, so please make sure you have a look at that. By attending this webinar, you are agreeing to abide by that code of conduct. There are two ways of communicating with us during the webinar. Feel free to use the chat function. You can either use the chat to contact the host directly or everyone. But please, be aware if you put anything in the chat function that everybody can read that's personal, that we can all see what that is, and above all, obviously be respectful to everyone that's here. However, if you want to ask a question for us to pick up and ask one of the speakers, please use the Q&A function for that, so that we can that the questions for the speakers don't get lost within the chat function. So please type your questions, type them as you think of them, don't wait till the end, because then we can keep an eye on them coming in. So as you think of a question, put them into the Q&A, and hopefully we'll have time for the speaker to answer them all. We also have live captioning for the talks today, so if you want to turn on the captioning on or off at any point, if you want to use it or don't want to, you can do that using live transcription button, or sometimes that shows up as a CC button at the bottom of the Zoom app. We're really grateful to our transcriber, One-to-One Caption, for captioning these talks today. So they're also being recorded and should be available in the next day or two on the Tigers YouTube channel, so please also be aware of that as we're going through. If there's anything that you don't want to say that want to be recorded, then please don't. The last thing I'd like to say before our first speaker is that this is a free event, with people putting their time into this, and the speakers coming along to give you these talks, but the Tigers support the Cowrie Scholarship Foundation, which wants to fund 100 disadvantaged Black British students through UK universities in the next decade. So please consider donating to them. If you do donate to them, please put Tigers in the comment section of their donation, of your donation, sorry. Okay, so I think that's all the housekeeping, but I'm sure if I've forgotten anything, someone will let me know, but just watch the chat in case I've missed anything. So on to the main event. So I would like to introduce our first speaker this afternoon, who is Mohamed Katish, who is going to be talking to us about, well, the title of his talk is Minority Making Major Contributions in Renewable Energy. Mohamed is a 50-year chemistry student pursuing a Masters of Chemistry at the University of Edinburgh. So I am going to pass over to him now. So Mohamed, take it away.
Hi guys, good afternoon. My name is Mohamed Katish. Would you like me to start presenting my work just now, Professor Catherine?
Yes, please share your screen.
Yeah, we'll do.
That's it.
Right, can you see it? Yeah.
Yeah, that's it.
Yeah, so good afternoon, guys. My name is Mohamed Katish. Today I would like to talk to you about my industrial placement project with Sunamp, a project titled Phase Change Material for Thermal Energy Storage Applications between 70 and 120 Celsius. First, let's start by talking about thermal energy storage. Thermal energy storage is the phenomena where thermal energy is stored in a medium to be accessed at a later point for different applications, such as water heating and power generation. Thermal energy storage is very important because almost 50% of the world energy consumption goes towards heat. Also, thermal energy storage can play a vital role in optimizing renewable energy sources by bridging the typical the gap between the supply and demand. There are different types of thermal energy storage, one of which is latent heat storage. Latent heat storage is where a material is heated until it changes phase, and when the material reaches its phase change temperature, it will absorb large amount of heat. This heat can be recovered when the material reverses phase. A material that undergoes such a change is called phase change material, or just PCM. There are many classes of PCMs, organic and inorganic. These classes vary in their melting point temperature and then latent heat available in them. Many companies utilize latent heat storage technology to produce latent heat devices. Companies like Sunamp produce latent heat batteries and hand warmers. Sunamp was working to develop a medium temperature heat battery for the temperature range 70 and 120 Celsius. For this heat battery, Sunamp chosen trimethylethane, just TME, as a PCM. TME is a versatile polyol with significant quality features. Its trihydroxide content gives it extra stability. TME has a solid phase transition at 81 Celsius, where it changes from low symmetry alpha phase to highly distorted gamma phase. TME has high latent heat of 190 kilojoule per kilograms. A heat battery with TME can be very viable in heat recovery applications and commercial hot water heating. However, producing a heat battery with TME can be very challenging and time-consuming because TME has high melting point of around 180 Celsius and high vapor pressure, which makes it sublime at around 135 Celsius. As you can see here, the sublimation is quite intense and it could expose the lab staff and lab technicians to some occupational hazard. So the aim of the project was to overcome the issues associated with the manufacturing or the production of medium temperature heat battery. And the approach that I took was by finding an additive which will reduce the melting point of TME and inhibit its sublimation. So I screened many additives, the TME mixture additives, and only one additive showed great potential. Is additive number 2, TME mixture with additive number 2 melted the temperature below 100 Celsius and did not sublime. And to understand the effect of adding additive to TME, I screened a wide spectrum of TME-Additive2 mixtures, and what I found is that the addition of additive 2 to TME reduces its melting point until it reaches a diversion around 45% of additive 2 added. And I also found that the addition, the sublimation of TME will only be inhibited if 48% of additive 2 added or above. So it's more like colligative properties. I have used DSC, differential scattering chrometry, to measure the or to compare the latent heat available in TME-Additive2 mixtures and with the pure TME sample. And what I found is that the TME additive mixture has two-phase transition, which both have the more latent heat available in them than that of pure TME. And also on the cooling curve, notice that the TME-Additive mixture subcools. Now, subcooling is the phenomenon where a material crystallizes below its freezing point. And this results in inefficient recovery of energy. Fortunately, subcooling could be inhibited by using nucleators. So at the end, the conclusion that the new formulation has 3 major advantages. It has low melting point, does not sublime, and it has higher energy output than the pure TME. However, there are a few things that still need some work. For example, the subcooling issue we discussed earlier, and still need to find a nucleator for this. Studying the chemical stability of the mixture, which will give us an opinion of or tells us about the life, the expected life from the heat battery. And finally, studying the structure and the identity of the mixture using structural and spectroscopic techniques. At the end, I would like to thank my industrial supervisor, Dr. Odlin, and my academic supervisor, Professor Pullum, for their support throughout this project. And I also would like to thank Tiger and STEM team for giving me the opportunity to present my work at this seminar. Thank you. And any questions?
Thank you, Mohamed. Yeah, we've got a couple of questions here for you. The first one's from Lorty McDonald. So where does the stability of the TME stem from?
The stability of TME stems from its structure. So the central carbon atom in TME is beta to the 3 hydroxide groups, and it does not have any hydrogens. So the absence of beta hydrogen atoms blocks any elimination processes. That makes sense, yeah.
Okay. You have another question here from Madison Denholm. Have you considered an engineering solution to improve the preparation procedure?
Yeah, that's a good question. Yeah, an engineering solution could overcome like the manufacturing process, but the main challenge is like when leaving the heat battery with the customer. If the heat battery casing gets damaged and then the customer continues to use it without noticing the damage, the TME will still sublime and make its way to the customer lungs. You know, so that's why we went for a chemical solution instead of engineering. Yeah.
I saw at the end that you perhaps used some spectroscopy techniques. So as a spectroscopist, I was just going to ask what kind of approaches you would maybe use for your characterization.
Yeah, so currently I'm working on the nucleator aspect and the stability, but for spectroscopy measurements, I was thinking of doing an infrared measurements, and I was thinking of maybe selecting a sensitive indicator group, OH group, and like measuring the band over a temperature gradient, and then this could tell me about the structure of the mixture.
Yeah. Okay. I'm going to ask you one more question, which I guess is a more personal question. But how are you enjoying your project? Obviously, I guess that's been influenced by, you know, COVID, etc. How have you enjoyed? I guess it's been a bit of a different experience from what you might have expected.
Yeah, it's quite different, but I think the guys at Sunamp like did a great job in making us comfortable and, you know, making sure that everything is like normal. Yeah.
Okay, thank you very much, Mohamed. I really enjoyed your talk.
Thanks, Professor Catherine. Right.
Could you stop sharing your screen?
Yeah, I'll do now.
Claire can.
Hello.
Hi, Claire. You're there. I couldn't see you there. So our next speaker is Claire Z, who is currently doing an undergraduate degree at Imperial College London with support from the Agency for Science, Technology and Research, which I guess we all know as A-STAR. for the National Science Scholarship of Singapore. So she's going to talk to us today about her unexpected encounter with computational chemistry. Can you share your screen?
Yes, let me share my screen. Can you see the screen?
Yeah, you're good.
Okay.
Please take it away, Claire.
So hi everyone, my name is Claire, and today I'll be sharing with you my unexpected encounter with computational chemistry. So firstly, I would like to talk about how I got into computational chemistry and then introduce to you guys two main methods used, mainly quantum mechanics and molecular dynamics, and then go into briefly two projects I've done so far and my future plans. So before and at the start of university, I spent a lot of time working in the lab, synthesizing nanoparticles or doing metal recycling. But since COVID pandemic kind of prevented laboratory projects from taking place, I started to think about how can I still work on science, which I'm very interested in, and the best way to do that is to take this computationally. So I started to approaching professors and asking for opportunities. And I've worked last year on a Europe project with Professor Patricia Hunt and Professor Tom Welton from Imperial and University of Wellington, New Zealand. So before going into the specific of the project, I'd like to give you a brief introduction to computational methods. So in computational chemistry, there's mainly two methods. Firstly, it's quantum chemical methods, which is basically based on quantum mechanics and calculate energy and electronic structures in the molecular system through solving the Schrodinger equation. It is quite versatile, but the solution is usually accuracy of the solutions based on approximation use. And because of the high demand and intensity, it's usually generally used on smaller molecular systems because it can be quite computationally expensive. But for larger systems, such as, for example, composite proteins or membranes and lipids, we can use molecular mechanics and dynamics to simulate them. And this is usually using classical physics to model atoms as like strings and bonds. And we use empirically or experimentally determined parameters to calculate force fields to represent the potential energy surface of the system. So for my project last summer, I worked on the solubility of ionic liquids using tensional functional theory. It's one of the computational methods using quantum chemistry. So ionic liquids are salt that are liquid at room temperature. So they are characterized by low melting points and high thermal stability and high electroconductivity, which makes them quite ideal for electrochemical sensors. So there's quite a lot of intense interest about studying them and studying the solubility of different organic solutes into them, but the specific interactions is actually quite elusive and not really well understood. So my project is aimed to use theoretical calculations and modeling to understand the specific identified interactions and how strongly the solvation energy and the association energies are. So this is two organic solutes that I study in my project, BZCC and BZCN. So as you can see, there's a benzene ring. And for the BZCC, there's two quite nonpolar alkyne groups at the end. While for BZCN, there's a cyanide group on both ends, which makes it a little bit more charge polarized. And this is the ionic liquids with the cation and anion used in my studies. So to study solvation, there's actually quite a lot of models. There's explicit solvation, which is putting in cations and anions of the solute explicitly next to the molecule to model solvation, while implicit implies using a dielectric continuum to simulate a homogeneous medium around it, which is usually less intensive. So today I would like to share with you about explicit solvation, which is by putting in the molecules right next to each other. But firstly, my project optimizes each of the solutes, the BZCC and CN, and the cation and anion in gas phase, and calculated the electrostatic potential. And as you can see, for the BZCN with the cyanide group, the charge is more polarized with the negative charge mostly on the cyanide group and the cation and the positive charge on the ends of the ring aromatic ring hydrogens. Well, for the BZCC, the charge distribution is much less extreme as you can see in comparison. Well, for the cation anions, the charge, well, the negative charge is mostly concentrated on the sulfur side of the anion. And the cation positive charge is mostly on the hydrogens, as you can see on the center ring. So with these ideas of how the charge is distributed, it's possible to place place the cation and anion next to each other and conducted lots of gas phase studies. And the resultant geometries can be ranked by their association energy. So for the BZCC, as you can tell, there's actually the strongest stabilized geometries based on their pi stacking. And when they're originate from the aromatic ring, and as they flips into a different geometry, the energy of association becomes less negative. But for BZCN, the cyanide group and the hydrogen on this carbon atom, which correspond to a hydrogen bonding, actually makes this geometry more stabilized. And while the pi stacking between the BZCN and the cation is actually less stable and higher in energy. Well, for the two solutes and the anions, both of them are stabilized by hydrogen bonding. As you can see, they have a preference of having the anion next to the aromatic ring, which is thought to be contributed by a two hydrogen bonding possible between the aromatic ring hydrogen and the salt and the oxygen on the anion. And this geometry is also observed in the BZCN solute ion clusters. And with these findings, we can actually build put the cation and anions together to build a bigger cluster, and also add in the implicit solvation to study it further. So this is these calculations give us actually a good understanding of how what is actually happening in the solvation and what forces are contributing to stability, which is quite useful. So I'm currently working in my second project on lipid force field characterization using an EMBER MD. So EMBER is a molecular dynamics package. So in this project, I studied using molecular dynamics to simulate membrane bilayers and which is composed of individual lipid molecules. So, So in my start of my project, I'm using gas phase and solvation calculations to derive charge parameters for the specific PIPS molecule, which is phospholynyl phosphates, and deriving charge parameters for the force field, which allows for the MD simulation of the lipid bilayers. So from doing these computational projects, what taught me is it's quite important to try new things, because on reflecting back, actually at the start, I didn't have much experiences with computational chemistry, and I thought I didn't have the ability to master them due to lack of experience. But then I find with doing these, it becomes easier with experience and practice. And actually, I find it quite enjoyable, and started to actually appreciate the flexibility and intellectual stimulation that comes with theoretical work. And if there's one learning point I want to share with you guys, is that it's always try new things and be confident in your ability to master new skills. And that is quite enriching for me, which was unexpected when I first started. So encouraged by this experience, I'm currently planning on taking a year before continuing with my PhD study to work as a research engineer back in A*STAR. So I'll be working on combining computational chemistry with experimental work, actually, to develop electronical materials. So there's an automatic synthesis platform where I'll be using calculations to direct and to try to develop new materials. So I'm quite looking forward to that. So I would like to thank my supervisors, Professor Tom Welton, Professor Trisha Hunt, Professor Ian Goh, and Julian Becker for their valuable guidance and feedback throughout the project. And thank you for your attention, and hope you enjoyed the presentation.
Thank you, Claire. I really enjoyed your presentation, and I really liked that sentiment at the end about trying new things. And I think sometimes it's important to try things that also terrify you a little bit, because you learn a lot from it. So I guess taken from that, obviously I guess you would have preferred to have done a lab-based project, but the circumstances have changed it. Did you actually want to do a computational project to start with to, you know, push yourself, or was it kind of, oh, I have to do this, but I'm going to embrace it?
To be honest, before the lockdown restrictions, I actually had a synthetic placement planned for next summer. So up until June, I was actually still like talking to my supervisor, can I still like work on it's actually for a drug discovery, making a new molecule. But then I started to think, like realistically, that's not going to happen. And at the start, in my first and second year, I actually find computational work a little bit difficult. So it's actually, I was like, I was a little bit, I guess a bit scared to start. But once someone, I guess initial supervisors, they're quite, they gave me quite a lot of guidance, and then started to realize actually learning these things is actually quite easy to pick up, especially computational skills. There's a lot of tutorials, there's a lot of guidance, and there's also a lot of time, because you're not really limited by being the lab resources and stuff, so you can learn and always ask questions, and Google is your best friend. So so after 1.5 years, it is really like, I find at the start, I say it was pretty an unexpected encounter, but at the end, I'm quite grateful for pushing me and picking up new skills.
Yeah, and I think it's given that advantage of the situation being where it is, you've actually been able to immerse yourself in learning to do it, rather than, you know, trying to do a little bit here and there where it's harder to pick up than actually having to throw yourself right into it. But I noticed that you've gone from ionic liquids to lipids, which are quite different projects in themselves. But was your main aim to study the systems or learn the different was your main aim to learn the different computational approaches for the two different systems, or to study the systems as well?
My first project is mainly based on quantum chemistry. So after learning that, I was trying to challenge myself and pick up new skills further. So my supervisor, Professor Ian Goh, he's working on molecular dynamics and force field prioritization. And when I look at the project, it's like, this is something it's quite useful, and a lot of people are working on it. And molecular dynamics is a really popular, I guess, method in computational chemistry. And I feel like I should use this opportunity to pick up this new skills, which could be quite useful in the future. Yeah. So I try to choose something that's different from what I've done before.
Yeah. Okay, thank you. I don't think we have any more questions for you, but that was great, and good luck with what comes next.
Thank you.
Where am I? So if you could stop. Yep. Back. So our next speaker is Jenny Zhang. Jenny completed her PhD in bioinorganic chemistry at the University of Sydney in Australia, with a brief stint at the Hebrew University of Jerusalem. She was awarded a Marie Curie Fellowship to work at the University of Cambridge with Professor Erwin Reisner to explore new biocatalysts can be exploited to generate solar cells, and she's now been awarded a BBSRC David Phillips Fellowship to start her own independent group. She has also recently been awarded the RSC Felix Franks Biotechnology Medal for her work, so she has a really outstanding early career track record. So I'm really looking forward to her talk, where she's going to take on telling us about the rewiring of photosynthesis to another level of complexity in living cells. So her talk title is Integrating world's natural and artificial photosynthesis. So Jenny, if we can see your talk, so I'll let you take it away.
Yeah, thank you so much, Karen. And thank you to the previous speakers as well. I really enjoyed listening to both your talks. And of course, thank you very much to the organizers for putting together this wonderful webinar series. I'm delighted to be involved and delighted to be here to tell you a little bit about what I do in the lab, which is trying to integrate two worlds together. I'm trying to integrate natural photosynthesis with artificial photosynthesis in the context of trying to find a new biohybrid method or approach for delivering renewable energy. But equally excited about telling you about that. I'm also excited about just telling you a little bit about my personal journey, how I got to be doing the unique research that I'm doing, because you see that it's not very common. And so perhaps the best place to start is just to tell you where I come from and where I am now. So I'm currently based in the UK in Cambridge. That's where my lab is based. But as mentioned by Karen, I did all my university as well, and I grew up in Australia. And so I get a lot of comments from people asking me like how I cope because of the big change in climate and you know, in different countries. But actually, all of that is nothing compared to my first migration experience, because actually I was born in China. In Guangzhou, and when I was 8, I migrated over with my mum. We migrated over to Australia, to a little town or city called Gosford in Australia, where we did not know one single word of English. We didn't have much money as well, so it was a complete restart, and so I had to relearn the language and the culture, and it was incredibly challenging. But in hindsight, it was also incredibly empowering and enriching, because it made me unafraid to enter into new worlds and explore new things. It made me realize that, you know, the power of integrating two cultures. There's more than one answer to everything. I basically created an identity not based on one single norm, so it was incredibly empowering. And so that's really reflected in my career trajectory. So you heard that I also went to Israel for a little bit, and that was a completely different way of doing science, as well as the way that chemistry was done. And so basically coming to UK is actually a piece of cake. And so in my research itself, you can also see that I really do like to enter new worlds. So in my PhD, I was very passionate about understanding anti-cancer agents. I was synthesizing them, investigated how they interacted with tumor models. And then for my postdoc, I decided I want to try something new. And so I completely changed, and I now am in the field of energy conversion, and I learned electrochemistry, and I learned how to interface materials with biological biocatalysts or enzymes, which I'll tell you about in a second. And now I'm driving my own research. I'm doing my own group is now pursuing biohybrid approaches for energy conversion. And I'm really making use of all the lessons and tool sets that I gained in my past to drive this very unique vision. So let me tell you about the science that I'm actually doing. So to understand the rest of my talk, I need to give you a little bit of introduction about photosynthesis. So we all know what photosynthetic organisms look like, right? So we know about plants, and some of us also know about algae and cyanobacteria, which are photosynthetic bacteria that live in ponds and water. And this is the equation that we typically get taught in school, which represents photosynthesis. But actually in real life, photosynthesis is much more nuanced and complex and interesting than that, because if you zoom into all the photosynthetic organisms, what they have in common is that in one of their organelles called a thylakoid and the membranes, you will find the photosynthetic electron transport chain, which is the cartoon, a very simplistic cartoon is depicted here. And everything starts with photosystem 2. This is this enzyme here, PS2. And PS2's role is really to absorb sunlight and use that energy to extract electrons from water, breaking it up into, you can see here, oxygen and protons. And those electrons are passed along this chain here and eventually ends up in photosystem 1. And in photosystem 1, what the role of this protein is to inject, harvest more sunlight and convert that energy to electrical energy, powering the electrons that's passing through here, so that these electrons, these energized electrons, can be picked up by another shuttle, in this case, ferredoxin, FD. And this ferredoxin shuttles the electrons to different pathways depending on the needs of the cell. So it's not just the one pathway. And of course, the most well-known pathway is the one, the Kelvin-Benson cycle. It goes through Rubisco. This is where carbohydrates form, biomass is formed. And in the context of energy, photosynthesis is really photosynthesis is really providing our energy through this pathway right now. So 12.6% of our electricity in the UK is derived from the burning of biomass to generate electricity, but also the biomass can be taken and fermented to form biofuels as well. So this is the pathway in which current technology is using photosynthesis. So the main thing, though, I want you to get out of the slide is that photosynthesis is complex, but actually individually there's lots of very, so I'd already talked about photosystem 2 and 1, but there's also other enzymes. Individually, there's some very impressive components and biomachineries that can do photo to chemical conversion extremely efficiently. However, you're going to hear from some people who will try to convince you that photosynthesis is actually very inefficient, but they're also correct, because it really depends on which part of photosynthesis you're looking at. So if you're looking at the entire pathway from the start to the end where you get biomass burning, yes, it's very, very inefficient. But if you zoom in on the individual components and parts of the pathway, it's actually quite impressively efficient. And so, as chemists and material scientists and physicists, we've given ourselves a goal for the last century, and this is one of the holy grails in chemistry, and that is to beat photosynthesis. So essentially using synthetic materials to completely replace the components of photosynthesis so that we can produce fuels from sunlight. And so you can see here several quotes, how can we do this? We should do this, but just faster, cheaper, better than nature. And even Bill Gates jumped in on the bandwagon a few years ago and wrote a piece about it and how wonderful this is going. And so this is the field known as artificial photosynthesis. And so you can see here, this is one of the first artificial leaps, which can essentially do water splitting, like using purely synthetic materials. And so this field is actually quite progressing very, very well. We have at our fingertips all these synthetic light harvesters that are much more efficient and much better at harvesting sunlight than leaves can, than natural pigments can. And the other thing that we have going for in this field is that it has very straightforward wiring, because you can see here I have a very simple 2-electrode system. Essentially, you just need 2 components. One of the electrodes will be for oxidation, so we can extract electrons from a feedstock. Those electrons will be taken to another electrode where it can do reduction reactions. And so that's where the fuel forming comes into play. And all of this should be driven by sunlight. And you can see that the wiring is so simple that you can sometimes just integrate it all into just the one integrated sheet. Now, to basically break bonds and reform bonds quickly. And if we want to make very simple chemicals like hydrogen, that's fine, we have some pretty good catalysts. But if we want to make something more complicated, fuels, for example, carbon-carbon bonds need to be made, CO2 reduction, for example, then we need to look at, well, actually nature has better catalysts for those. So we're looking at catalysts that are scalable, that are efficient, that can last a long time. And so basically in the lab, we are unable to make these catalysts, at least not yet, and probably be very challenging thing to eventually develop. So basically this is where we come to Can we basically, instead of working individually, can we combine the strengths of natural and artificial photosynthesis and kind of cherry pick what we like from each system and combine them so that we have a new hybrid system, so a semi-artificial system. So for example, can we take the catalyst and use the wiring regimes from artificial photosynthesis so that we can do overall solar to fuel formation or solar energy conversion? And if so, what are the challenges? How do we do that? And so this is kind of the work that I started doing in my postdoc. So I'll tell you a little bit about that. There are obviously, you can imagine, lots of challenges. So to start us off, understanding how we would even, approach such an approach, we look at what, this is the biocatalyst that we want to wire to an electrode. Okay, so we need to understand that first. You can see it's quite a big enzyme in terms of biocatalyst goes, one of the bigger ones. I already talked about how it's very important for kickstarting photosynthesis. This is photosystem 2, and it's very inspirational in its ability to convert sunlight into chemical energy. So how does it work if we strip it all back? What it contains are these pigments called chlorophylls. So basically they harness energy, and those energy are transferred to what we call a reaction center. And the reaction center is where the charge separation occurs, and it provides a driving force to pump electrons from one side after oxidation happens, or oxidation of water happens, to the other side. So electrons are delivered on the other side of the protein, and we basically want to collect it using an electrode. And so we can use these electrons then to study the functionality of the enzyme, but also to perhaps use them for power generation or to make new bonds for chemical formation. So a very easy way, well, I wouldn't say it's easy, a very straightforward way of understanding and studying how this could be wired to an electrode is to perform what we call chronoamperometry. So this is an electrochemical method where essentially we keep the electrode at one constant energy level. We shine light on the electrode, and you can see that under light we have this spike in current, and this spike in current corresponds to the water oxidation, that is the reaction that's occurring. And so the magnitude of this spike basically tells us about how active our catalyst is. It tells us about how well the catalyst is wired to our electrode, and also tells us about how many catalysts we have on our electrode. So we actually can find out a lot of useful information by doing this one technique. And just to show you an example how we can characterize the enzymes. So here again, I just show you the electrochemical setup. It's a 3-electrode system. It's just a common analytical tool that we use. And so if we can wire these proteins to our electrodes, here we show a comparison. Okay, so we have photosystem 2 derived from 2 different organisms. One is from spinach, the spinach you can get from the supermarket, and one is from a thermophilic cyanobacteria. And here we basically apply different energy levels on our electrode, and then we look for the photocurrent spike. And you can already see that the photosystem 2 derived from the thermophilic cyanobacteria is much more active than the spinach one. And so using this type of plot by, for example, assessing at which energy level we first start seeing the spike, we can determine how efficient also our catalyst is. And also we can learn a whole lot of different other information. For example, we can see whether there's recombinant pathways where the charge would recombine with each other. But I won't talk about that because we won't have enough time. But essentially, I just want to communicate that this is a very good and easy method of looking at the wiring. Now, so this is a really good way of studying the functionality of the enzyme. However, if we want to use them in devices, we really need this photocurrent to be huge. Okay, we need it to be almost like a milliampere per centimeter squared for it to be able to drive devices. So we can do lots of little tricks to really enhance this spike. So for example, we can add in synthetic electron mediators. So they could be like quinones, for example, and they're diffusional. So what that means is that we can have these enzymes, and they can be oriented in any way. They don't have to be directly facing the electrode because it'd be very difficult to always orient them the right way. And that way we can use these mediators to help shuttle electrons wherever the photosystem 2 might be distributed near the surface of the electrode. Another way of increasing the photocurrent is actually through some engineering. And so here you can see this is the photocurrent output over time from photosystems or electrodes. And in the initial stages, we had very low output. And then in 2015-16, a member of my research group and Owen Rice's group, this is work done by a PhD student, Dirk Mersch, she redesigned the electrode. And basically, it meant that we can improve the photocurrent by 3 orders of magnitude because of the fact that we are now able to have structures like a conductive sponge. We're able to sponge up, soak up all these photosynthetic enzymes and have them be wired and collecting electrons for us. And because of this feat, we're able now to basically create these devices where we're wiring up different enzymes with each other to do whichever fuel forming reaction we want. So this is an illustration of what one of the devices would look like. So you can see zoomed in into each of these electrodes, we have these conductive sponges, and we can load it up with photosystem 2 on one end, and on the other side it's very modular. We can put in, say for example, a hydrogenase so that we can do hydrogen formation. So this is what we call water splitting, and we know that hydrogen is a very green fuel that we want to create more of in the future. But also we can put other enzymes in, like for example formate dehydrogenase, and that can help us to reduce CO2 and create it basically from a different type of molecule. So in this case is formate. And so these are really exciting systems, and this is work that I haven't gone into great details of, but if you're interested, I've written a review about this in simpler terms, and you can investigate that further. So at this point, we're really excited, and we can make all these prototype devices. However, what was really limiting us with these devices was the fact that these are enzyme-based. And unfortunately, purified enzymes are intrinsically unstable because they just break down. So what we ultimately want to do is think about integrating instead living systems. So for example, can we wire enzymes onto electrodes while they're still inside of living systems and they're still being prepared, and so they won't, even if they break down, we can still wire to new ones. And so this is the type of research I'm really doing now. Okay, so I've already mentioned before that from the photosynthetic transfer pathway, this is where current bioenergy is being derived. Okay, that's what we're using right now. But if we can think about rewiring this pathway, so for example, we can collect electrons from an earlier point, we can bypass a lot of inefficient steps and perhaps create a more efficient and new type of bioenergy using living cells. This seems like it's quite almost a little bit sci-fi, but actually we have something very similar working already. So I don't know if, obviously last 2 years there haven't been any music festivals, but for those of you who have been to Glastonbury, you may have already seen these. Essentially, this is fantastic work that was done by a group in Bristol by Ioannis Eopoulos. So sorry if I didn't get the name quite right, but I really, really love this work where basically he's using microorganisms to break down waste and extract those electrons and use those electrons to power lighting and the recharging of phones. And how it works is that if you look at it again, this is a device, it's a 2-electrode device where we have on the anode, we have some microorganisms that can eat the organics, and again, like I said, you can derive the electrons out, and electrons are used to reduce something else on the other side, and that's where we get the power. So what we want to do is something similar. We don't want to use the same type of microbes. Instead, we want to use photosynthetic cells. These could be cyanobacteria, which I showed here, could be algae, and this is highly advantageous because of the scalability of these materials. You know, they're the most abundant materials in the world, and also they only require water, sunlight to give out electrons. And so this is a really appealing technology, as you can imagine. And also there's already prototypes that are working. Now, you can imagine that we already talked about some of the complexities of wiring isolated systems to electrodes. So when we come to wire whole cells to electrodes, you can imagine that the complexity multiplies. So I just want to show you briefly some of the challenges that we're facing. So you remember me complaining a little bit about how big the photosystems were? Well, these are even bigger because they're whole cells, so they're 2 orders of magnitude bigger. And so one of the challenges is to create suitable electrodes that we can wire to these cells. And these striated formations you can see here, those are the thylakoids. That's where all the enzymes are embedded. So we want to extract electrons from those organelles. So this is our first attempt. We use these porous electrodes. We just enlarge the pores, and you can see that we can have the cell sitting inside of the pores, and it turns the electrode green. The second challenge is that, well, you can probably guess this, extremely complex electron transfer mechanism. So if we kind of zoom in at the biomaterial interface here, where we want to collect electrons, you can see here we have the photosynthetic electron transport chain. And what we really want to do is redirect the electrons early on in the chain so that we can direct them into the electrode. Now, luckily, there is already a natural pathway that does this. It's called exoelectrogenesis. The problem is we don't understand why it happens, and we also don't understand how it happens. We just know that there is some sort of chemical that's taking electrons out and dumping it outside of the cells. And so without understanding what this mechanism is and what is this X molecule, it's very difficult for us to rationally design systems to improve this pathway, to improve this rewiring pathway. So this is actually one of the challenges that we're facing. So once we understand this better, we hope that we can redesign X so that we can use our chemical tool sets, etc., and basically really enhance this rewired pathway. Okay, so another challenge is the fact that now before you could see that we had these very straightforward profiles. Now with living organisms, we actually see these very complex photocurrent profiles where we get dips and peaks. And again, we don't really understand why this happens. And we know that they change with different organisms and also in response to different stresses. But obviously this is reflecting something about the journey of the electrons, and we really need theoretical modeling to help us untangle it. So this is something we're also working on right now. But the last thing I want to show you is, and you probably saw that from the last slide, is that the challenge is that we now have lower photocurrent, but the photocurrent is sustained. So here's a summary of that. This is what we call turnover number on the y-axis. This is just the accumulated product per catalyst. And this is over time, over 5 simulated days. So that's why we have light on and off. And you can see that the isolated system, isolated photosystem 2 on electrodes, give really a lot of products really quickly, but then it dies. With living cells, cyanobacteria, it basically goes on and on and on until I stopped it, but obviously it was very happy and healthy, but it's much lower. The rate of product production is much lower. However, promisingly, if we can add an electron mediator, we can really change the kinetics. So we can basically show that we can rewire this pathway. We just need to improve on our electron mediator. So this is definitely another future challenge. So I just want to give you a gist of some of the challenges we have. And obviously, we need a lot of different approaches to solve this. And this is actually to my delight because, like I said, I love basically integrating different worlds and different worlds of knowledge. So we need engineering, we need different types of chemistry, we need physicists to help us figure out the photophysics, we need mathematicians, we need biologists, obviously. And so it's a really exciting space to work at. And I guess my message is that for anyone who also, like me, has a very unconventional background, who is used to navigating different worlds and integrating different worlds, who basically who feel that maybe they can use their difference in a way that is productive, then science is a really, really exciting time to be getting to science, just because of the fact that we really need interdisciplinary, multidisciplinary efforts. There's no problem in the world right now that would be solved by one technique alone, and we really need people who can navigate different worlds comfortably. So really use that as a superpower if that is indeed something that you possess. So hopefully I've given you a taste of what future biohybrids may look like for energy conversion. So we basically went from enzymes to living systems, and we're aiming for high stability and high efficiency. I talked a little bit about how this might apply to energy conversion and fuel generation, but it really is the basis of lots of other types of exciting technology, which I don't have time to go into. And if you're interested in something like this, the RSC and Chemical Science, we're having a symposium on this later this year, which I'm chairing. So please come join us there. It's really exciting. And with that, I think I just want to finish by thanking my team. So I have a fabulous team of engineers and biologists and chemists who work very productively together. And I'd like to also thank my collaborators from all different departments, as well as different parts of the world. Yeah, it's a really stimulating place to work at right now in science. So with that, thank you very much.
Thank you very much, Jenny. That was really nice analogy between your life journey and your career journey, and I totally agree with your sentiment that, you know, we do need multidisciplinary science and groupings to solve these really, really big problems. That was really amazing science that you're doing there. So I'll start off with a question for you from Rachel Oliver. Is biocompatibility of the electrode materials an issue? Is there a risk that they break down or degrade or poison the living system, the organism? Sorry.
Yeah, that's a really great. That's a fantastic question. Thank you for the question. Actually, I mean, yes. But that's not a bad thing. And in fact, if it degrades, we want it to be able to degrade so that it's in a environmentally friendly manner, because nothing really lasts. And one of the good things about cyanobacteria is that they recycle themselves. So it'd be great if they can, when they do stop working, they can basically break down, be reabsorbed by the earth, and then maybe we can have a new system come in. I don't think that's a big problem. So we do have to choose the electrode material really carefully. And at the moment we have ITO, which is indium tin oxide. Obviously, that's not the most scalable material, even though that's being used in photovoltaics right now. We want to switch to something better. And so that's one of the main engineering issues we have. Basically, finding a material that could either last, if not last, then be environmentally friendly if it does degrade. And so we're looking at all sorts of materials right now to solve that issue.
Oh. Caroline has a question. I think she wants to ask herself, so I'll just let her come in.
Yeah, sorry, I'm abusing my powers. Panelist, the Q&A function, it seems, so I'm just butting in with the video. Fantastic talk. Thank you so much. I have a question. I love the end, what you talked about, the interdisciplinary aspect of the work, and I'm asking a bit as a physicist. Are you looking into optimizing the photophysics of it? Is there something you can play with the spectrum? Is the sun spectrum really the most efficient way? And also, if you can comment on a curiosity, I've heard that maybe tunneling effects play a role in photosynthesis. That would be fantastic.
Oh, great. Yeah, so for physics part, definitely. So one of the major limitations in photosynthesis in terms of efficiency is that it's the photophysics itself. So it can't harvest half the sunlight, half the wavelengths that's within sunlight. And so one way we can use physics is to, well, first of all, use it to study how we can incorporate artificial or maybe not even artificial, we can maybe engineer something, bioengineer something, but basically another pigment or photosensitizer, which can help us to harvest more of the sunlight. And we really do need physical tools to help us do that. But in fact, actually, there's even though we we know a lot about photosynthesis, but there's a lot of gaps. So there's still a lot of room for biophysicists to develop tools to understand different parts of the pathway so that we can understand the final output. So we all have been concentrating on understanding biomass formation, which is great, but as I told you, there's many different pathways actually. And so we need biophysics to help us develop that. In terms of tunneling, I'm guessing you mean electron tunneling? Is that the type of tunneling you're talking about?
Sorry, I was actually meaning photon tunneling. I think when you said absorbed at some point of the complex molecule and they're going to the reaction site, I think I had heard that maybe tunneling effect.
Oh, I didn't say tunneling, but that's a really fascinating. I'm sure there's lots of very interesting effects going on there, but I don't think tunneling was one of those. So I'm sorry if I did misspeak there. But there is lots of very interesting biophysics to be understood in how photosynthesis systems are able to almost with 100% efficiency capture the light and transfer that on to create chemical energy. And one of those explanations is actually the occurrence of coherence, but I'm going to misspeak if I speak any further because I actually haven't looked into that much further, but there's a lot of quantum coherence coupling, I think, that goes on to perhaps, so there's a lot of quantum effects that explains why it's so efficient at those reaction centers.
Thank you.
Thank you. I was just going to ask something. There's another question here for you, but it follows on because it just strikes me how do you keep the cells, the bacteria, happy within the system to ensure, you know, that they're at the correct temperature, right? You know.
Ah, yeah, very good question.
Because I guess if they get unhappy, it's going to affect the pathway, and they're.
Yeah. Well, it's a very good question. At the moment, we're using monoculture to be, you know, clean with our result, and so we always just use the one condition so that we can understand one pathway very cleanly before we move on to more complex systems. But in nature, they're incredibly robust, and they can survive on glaciers. I mean, obviously, different species, but they can live in deserts. They can. It's just so diverse in terms of the habitats that they can thrive in. It's quite incredible, and they also love to live in cold culture, so with other species as well, and that's how they basically keep an equilibrium. But for scientific reasons, we basically keep everything at one condition.
Yeah, I guess that's the thing I was thinking all the way through, is how hard it is to recreate what nature does.
Yeah,
by itself quite easily. But I guess following on from that, Francesca's asked, she says, great talk, thank you very much. So what are the challenges of being able to scale up this type of technology, and how difficult will it be to get this to a scale where it makes a substantial difference to the world's fuel needs? That's a big question.
Yeah, could you ask the first part again? I think I missed.
Yeah, so what are the challenges of scaling up?
Scaling up. Okay, yeah, that's a really great question. So from our side, I mean, there's lots of engineers who are working on trying to make this scalable, and there's lots of little tricks you can play to, you can wire them in different configurations. But from my side, I think the major challenge is actually on the fundamental level. So I already explained to you some of the challenges that we have. We don't actually understand how electrons are coming out. Without understanding that, it's really hard to rationally enhance it. And so I think the main challenge is actually understanding. So all of this is quite new. It's quite, we only discovered that cyanobacteria can do this, just over a decade ago. So everything is quite new. So figuring out the answers to some of these fundamental questions will be number one in my head for how to even approach to get to scalability. But once we understand that, and let's say that we can get to a point, and we have done theoretical calculations, so we know, what numbers are possible, at which point will we outcompete current technology for bioenergy conversion. We know all of that, but we just need to meet those numbers. And so again, understanding the fundamentals will help us to rationally meet those numbers. At the moment, we're still about 1 or 2, with recent development, we're about 1 order of magnitude off. So yeah, so understanding the fundamentals, and then once we get those numbers, it'll be an engineering challenge where we need to find, okay, maybe more suitable materials, and figure out what kind of configuration we would need to really push the efficiency of these systems.
Okay, thank you very much. So we have to finish now. We've gone over by 5 minutes, so that's my fault. So thank you very much, Jenny, and thank you to our other speakers. It's been really inspiring to see some really nice science going on by our early career researchers, and as well as Jenny, who's moving into a more established career maybe. So I'm going to pass back to Sean, who's got some closing remarks, I think.
Thanks very much. I'd like to say thank you to all the speakers, as well as to our chair, Professor Folds. I'd like to announce that the next webinar in the series is in 2 weeks' time. That's on the 16th. And this will feature talks by Tihana Stefanic on forever chemicals, the magnitude, dangers, and future of PFAS compounds, as well as Livia Mascus on quantum effects in biology. Our keynote talk will be from Jamie Pitts on transitioning, cross-linking, and kinases, a PhD student's perspective. And you can sign up now via Eventbrite. The link should be in the chat in just a moment. In the meantime, we would also be very grateful if you could take a couple of minutes to fill out our short evaluation form. One of our co-hosts will post a link in the chat any minute now for this as well. Your feedback will help us to run the best events we can and potentially secure funding for more events in the future. So please do fill in the survey if you can. With that, I would like to just say thanks, and it's the start of Pride Month, so I'd also like to just say Happy Pride to everyone. And it's a good opportunity to remember that when we stand together and work together, that so much good things can take place and lead us toward a better future. Thanks very much, and see you all at the next webinar.